My name is Chandler Warr, chemical engineering major, do bioprinting research. So it's 3D printing, but instead of using plastic, you use a syringe with cells in a bioink. And the whole goal behind it, the long-term goal, is to make artificial tissues, artificial organs, like replacement part type stuff. That's a long ways off. I mean, it's just kind of like how an inkjet printer would work. And you just use a couple different simple colors, and you can create any picture that you want. So the same way, if you use a couple different bioinks, a couple different cells in there, a couple different materials, then you can print these 2D layers that are almost whatever you want. And then you stack these layers up like 3D printing does, and you've got a tissue and organ, you've got a kidney or heart or whatever it may be. What I'm trying to do right now is develop 3D cell culture with bioprinting. So 3D cell culture, in cell culture right now, we only do it in 2 dimensions. So it's a flat single layer of cells, which it's worked well for a long time. But cells in 2D don't always act the same as they do in 3D. And they have neighbors above and below and everywhere around them. And when you're doing different testing, drug testing for instance, you need to make sure that the cells that you're testing react the same as that they're going to react to the body. But you can't recreate the 3D cellular environment, that 3D cell culture inside, like in the lab right now. You can't do it, not reliably at least. And part of the reason you can't do it is because you can't get the plumbing right. You can't get, like in nature, cells are fed by a blood vessel system. So it's a plumbing system that goes really, really small. Right now in 3D cell culture, you're essentially left with cell clumps, cell spheres, circles. They can't get very big, or the cells in the middle die because they don't have the plumbing, they can't get a nutrient waste exchange. Where we're trying to use bioprinting, where you can create these 2D layers, where you can create plumbing and empty spaces within these 2D layers and then build this up. And you can get that plumbing next to all the cells that you're laying down inside the structure at the same time. So you build that up layer by layer and get a 3D cell culture, and eventually, like I said, that should be, and will hopefully be able to be a way to manufacture tissues and organs. Because a good 3D cell culture that's matured, has the right plumbing, the right vasculature inside it, is basically a tissue, it's an organ. That's almost the definition of it. So that's what I'm working on right now, 3D cell culture with bioprinting.